My name is Marcus Pereira, Assistant Professor of Infectious Diseases here at Columbia Presbyterian Medical Center in New York. I'll be talking to you today about updates on COVID-19 vaccine. These are my disclosures, and I'll be discussing investigational therapies during this presentation. This is the outline of my talk. I will be briefly discussing the types of vaccines and then going into a lot of data on the recent publications on COVID-19, as well as CDC recommendations and summarizing the variants for SARS-CoV-2 at the end of this talk. So going right into it, there are several different types of vaccines that are currently in use for immunization of adults and children around the world. So the one that is most commonly used would be live attenuated, and examples of that would be the MMR and varicella vaccines. There are inactivated vaccines in which the either virus or bacteria are rendered inactive and can no longer cause the actual infection, but are strong enough to cause an immune reaction. So those would be the inactivated polio and influenza. Now, a very common type of vaccine would be the subunit or recombinant or polysaccharide or a conjugate vaccines. Those are all operate very similarly, in which you combine the actual antigen of choice and usually a protein and a surface protein at that, and elicit an immune response to that. So common examples would be the hepatitis B, the pneumococcal, meningococcal, and the shingles vaccine, as well as the Tdap, in particular the acellular pertussis. Toxoid vaccines are usually just weakened toxins. So the tetanus vaccine, for example, is a prime example of that vaccine. And then nucleic acid vaccines would be the newer examples. These are mRNA, DNA vaccines, as well as viral vector vaccines. We'll be talking about those in a little bit. For COVID-19, there have been a number of different vaccine strategies that have been chosen to immunize individuals against SARS-CoV-2. So a number of candidates actually have used live attenuated vaccines. None of those are approved yet. Inactivated virus or SARS-CoV-2 have been approved in China as well as India. And then recently, Novovax, for example, has been a prime example of the protein vaccine for SARS-CoV-2. Now, the ones that we'll be talking the most will be this newer technology of the nucleic acid vaccines, both the mRNA vaccines and the Pfizer and the Moderna, which have been approved in the United States, as well as the viral vector vaccines, the Oxford or AstraZeneca, as well as the Johnson & Johnson or Janssen vaccines are great examples of those types of vaccines. A little bit about SARS-CoV-2, the structures and the spike protein, which we'll be talking a lot about. So many of you have seen sort of this three-dimensional render of SARS-CoV-2, sort of as a virus, and these sort of red dots are the spike proteins, which populate the surface of the virus. This is an electron microscope. You can see the spike proteins around the virus. This is a two-dimensional cartoon of SARS-CoV-2, and you can see the spike protein coming at the capsular membrane and the nuclear capsid, which entails the mRNA of the virus. So of particular importance to SARS-CoV-2 pathogenicity is the spike protein, which causes its binding to the ACE2 receptors in the surface of the cells, then causing the actual infection and the pathogenesis. So this is a rendition of the spike protein. It's actually quite a complex molecule. It's a trimer of 3 equal parts. And at the surface of this protein is the receptor binding domain, which is what actually attaches to the ACE2 receptor. This is where all the vaccine candidates currently approved, vaccine candidates and vaccines that have been approved target. It's the spike protein, in particular the receptor binding domain. So essentially all the vaccines have chosen to target the same antigen. So going right into the mRNA vaccines were the first ones approved, and we'll be talking about that data first. This is both the Pfizer and the Moderna vaccines. They are very similar in structure and in target. They are lipid nanoparticles that are formulated to enclose this modified mRNA, which encapsulates this nucleoside modified mRNA, which encodes a pre-fusion stabilized membrane anchored SARS-CoV-2, the full length of the spike protein. Essentially, there's mRNA encoding for the spike protein that is in the pre-fusion form, which is the one that you can actually neutralize with antibodies. And you have this lipid nanoparticle around that mRNA to stabilize it, protect it, and actually deliver the mRNA into the cells. And you can see in these two cartoons that were published along with the publications, you have the nanoparticle and the mRNA that basically delivers the mRNA into the cell. And then you have the transcription of the mRNA and producing the spike protein that then is on the cell surface, and then it can be recognized by the immune system. Both vaccines require 2 injections. The Pfizer is 21 days apart, the Moderna is 28 days apart. Interestingly, both of them, because mRNA is generally not as robust as DNA, they require deep freezing. The Pfizer requires minus 25-degree freezers, and the Moderna requires minus 20-degree freezers. These are Celsius, not Fahrenheit. And after thawing, they can last in the refrigerator anywhere between 5 and 30 days. But once at room temperature, you basically have 12 hours to actually inject the vaccine. So we'll start with the Pfizer vaccine, also known as BNT162b2 mRNA vaccine. This was a phase 3 publication on the New England Journal of Medicine in late December. So a little bit of word about the study designs of all of these vaccines. As part of Operations Warp Speed, one of the interesting aspects of that entire endeavor has been to really synchronize the study design and primary efficacy analysis. So all of these phase 3 trials encompass large numbers of individuals, including those with comorbidities. And safety analysis and outcomes were all very similar in that efficacy analysis was looking at PCR confirmed symptomatic COVID-19 after several days from full vaccination. And we'll go over each detail on the vaccines. So for the Pfizer vaccine, there were over 43,000 participants. All of them were over 16 years of age, 21,000 or a little over 21,000 with placebo and a little over 21,000 with the Pfizer vaccine. And there was a median follow-up of up to 2 months. HIV and other patients with comorbidities were included. The vast majority of participants were in the United States. Overall, this was found to be a very safe vaccine. And you can see here in the figures to the right, both at a younger age, 16 to 55 years of age, and those over 55, you can see that the systemic and localized side effects were quite similar and not that significant. They experienced fever, fatigue, headache, chills, muscle pains to a large extent, but largely to be expected. Now, there were some reports of anaphylaxis, and this was largely identified after approval or the emergency use authorization of this vaccine through the Vaccine Adverse Event Reporting System set up by the CDC and the FDA. They found 21 cases of anaphylaxis after almost 2 million doses, which was a rate of 11 cases per million doses. And 71% of those cases occurred within 15 minutes. Now, as far as efficacy, so again, defined as development of symptomatic and PCR-confirmed COVID-19 7 days after the second injection, the placebo arm had 162 cases, 9 of which were severe, and the Pfizer arm had 7 cases, 1 of which was severe. There were no deaths overall, but overall, this led to that 95% efficacy that many of us heard on the news back in December 2020. And here's a figure in terms of the cumulative incidence of cases in days after dose 1. So you can see that overall, the placebo arm in blue and the vaccinated arm in red, there is already a marked differentiation by day 14, day 21, from the first injection. You can see in sort of insert, this is just blowing up into greater detail, 3 days, 6 days, 9 days. And by day 12, you start seeing a separation in those developing cases and those not between the vaccinated group and the group not vaccinated. So all around, remarkable results, both in terms of safety, but also in particular in terms of efficacy, even starting with the first dose from this vaccine. So these were celebrated results and it led to a quick approval of this vaccine in the United States. In terms of the Mrna vaccine, also known as the mRNA 1273, very similar study design, 30,000 volunteers. They were over 18 years of age or older. There were 15,000 participants or so in each group with a median follow-up of also 2 months. They included several comorbidities, including HIV, liver disease, CKD, diabetes, and lung disease. And this was a trial exclusively in the United States. Overall, once again, it was found to be a very safe vaccine. And you can see here the breakdown, again, in terms of local events and systemic events, that there was pain, but no erythema, swelling, or lymphadenopathy. And in terms of systemic events, some fever, headaches, fatigue, myalgias, and joint pains or arthralgias, but nothing significant that was picked up in terms of other adverse reactions during the study trials. Just like the Pfizer vaccine, however, post-approval through the vaccine adverse event reporting system, they did pick up cases of anaphylaxis, in particular after the first injection. And in this case, there were 10 cases after over 4 million doses, so leading to a rate of 2.5 cases per million doses. The onset occurred within 15 minutes as well, and there was no anaphylaxis-related deaths reported after the Mrna vaccine. And in terms of efficacy, again, similar primary outcomes, symptomatic COVID-19 after 14 days from the second dose. Placebo had 185 cases, 30 of which were severe, and 1 case led to actually death. And the Mrna vaccine, there were 11 cases, none of which were severe, and there were no deaths. Again, remarkable results in terms of vaccine efficacy, up to 94% efficacy, and this was translated across all the groups and ages. So you can see here this cumulative event rate figure, very similar to the Pfizer vaccine. This is days after randomization. Again, you have vaccine on day 1 and the second dose after day 28 for the Mrna vaccine. And you can see that even before the second dose, there is already a separation in terms of cases between those who received the vaccine and the placebo arm. Quite remarkable results again, and led to the approval of the Mrna vaccine. Now, moving on to different types of vaccines. So we talked about the mRNA vaccines. Another new technology that was utilized for COVID-19 are these adenovirus-based vaccines, or the viral vector vaccines. A little bit different than the mRNA, but there are some similarities as well. In fact, actually, there have been decades of research for these viral vector vaccines, and they're actually used in military recruits and dogs. And actually, in July of 2020, an adenovirus-based vaccine was approved in the United States for Ebola, actually. Didn't make the news necessarily because we were sort of paying attention to other things. There are actually many advantages to a viral vector and adenovirus-based vaccines. They are very easy and cheap to make. They're easy to store. The adenovirus itself is quite stable, and all it needs is just regular refrigeration for up to 3 months, and that adenovirus is quite stable. They can be delivered via many routes, unlike mRNA vaccines, which need to be intramuscular. These can be intramuscular, but also subcutaneous, can actually be inhaled even. So a lot of different groups are working on different technologies for these types of vaccines. They usually contain DNA as opposed to RNA. So DNA is actually more stable than RNA. So again, leading to sort of this ability to only be refrigerator and no deep freezer requirements for these types of vaccines. There are some drawbacks for the adenovirus vaccines in particular. Prior immunogenicity can be one. So many of us are exposed to the different types of adenovirus on a daily basis, even as part of sort of a community-acquired respiratory infections, conjunctivitis, and other forms of infection. So if you have prior immunogenicity, your ability to respond to vaccine might actually be diminished. So what many vaccine candidates have exploited are sort of these less usual adenovirus that we don't necessarily have prior immunogenicity, or even from adenovirus from different species. So for example, the Oxford AstraZeneca vaccine is actually a chimpanzee adenovirus, so we're not that exposed to it, and we don't necessarily have this erratic immune response to the adenovirus. And the Johnson & Johnson vaccine actually has this adenovirus 26, which again is a quite uncommon adenovirus. Now, to make it even safer, because you certainly don't want adenovirus replicating and causing its own set of symptoms, so most of these vaccines actually, they are engineered to actually not be able to replicate. These are sort of replication incompetent adenoviruses, in which they actually delete key early genes from the virus, sort of E1A and E1B, those are deleted. So that virus itself cannot replicate in any host. So this is a cartoon characterization of the vaccine. Here you have the adenovirus that is carrying DNA material of your choice to deliver the vaccine. And then once the adenovirus gets into the cells, actually it delivers the DNA, and then through transcription and translation, actually it produces whatever target you want. And in the case of the adenovirus vaccines for COVID-19, it's the spike protein as well. So then once the cell produces spike protein, it actually makes its way into the cell surface, ready to be recognized by the immune system and then elicit an immune response. In some ways, it's actually just like the mRNA vaccines. Instead of having a lipid nanoparticle to deliver the genetic material, the nucleic acid of your choice, this is an adenovirus. So once you sort of think about that, it's actually a very similar technology and overall concept of these vaccines. So on to the actual vaccines. The one that has made most of the news is the Oxford AstraZeneca vaccine called ChAdOx1 and COVID-19. Now, this is not approved in the United States, but still an important vaccine to discuss, as it actually will be the centerpiece of the worldwide vaccination led through COVAX. So the Oxford vaccine is a replication-incompetent chimpanzee adenovirus vector that expresses a stabilized spike protein. It's 2 doses, 4 to 12 weeks apart. In fact, actually, there have been many trials around the world with this vaccine, and the one that was published at The Lancet late last year included 4 blinded randomized controlled trials, 2 of which were carried in the United Kingdom, COV001, which was a phase 1 and 2, and COV002, which was a phase 2/3 trial. There was one in Brazil, COV003, which actually was a phase 3, including healthcare workers mainly, and one in South Africa called COV005, which was a phase 1/2 study. Now, you might have heard in the news about when the data came out that actually a subset in one of the UK trials, the COV002, in fact, received only half a dose on their first injection, which was deemed a low dose arm, and then a standard dose as the second injection. So this was actually a little bit of an error in the delivery of the study drug, but had interesting implications. We'll talk about that. But this is sort of the low dose, standard dose cohort. Now, in terms of safety, it was reported actually to be a very safe vaccine, but there was some discussion about a case of transverse myelitis, which was reported 14 days after the second injection of this vaccine. And the researchers and the safety board thought that possibly related to the vaccination. Now, keep in mind that this is one case among several thousand doses. There were 2 additional cases of transverse myelitis reported, but later determined not to be related to the vaccine. And here are the results of almost over 11,000 participants, which actually for the efficacy arm only included 2 of the studies, one in the UK and one in Brazil. So the primary outcome, again, was PCR-confirmed symptomatic COVID-19 14 days after the full vaccination schedule. And the overall efficacy of the study was 70.4%. So not too far from the mRNA vaccines, but certainly not 95% that we saw with those vaccines. Now, interestingly, the twist in this study results was that the vaccine efficacy was actually higher in the low dose, standard dose recipients, paradoxically, and it was 90% in that group, and 62% in the standard dose recipients, mostly in Brazil and parts of the UK as well. Interestingly, it was a little bit of a puzzling result, still yet to be determined what the significance of this is. But in terms of COVID-19 related hospitalizations, there were 2 cases in the vaccine arm and 16 cases in the placebo arm. Severe disease, there were none in the vaccine arm and 2 in the placebo arm. And an interesting actual outcome that was not included in other studies. Now, remember that the outcomes for these vaccines was actually symptomatic disease. There was no mention actually of asymptomatic acquisition of SARS-CoV-2, which has implications for overall transmission of the vaccine, of the virus. So, but a subset of participants in this study actually had weekly PCRs obtained, and you could see who actually became PCR positive, even among those who were asymptomatic. And actually, any PCR positive swabs, there was actually almost a 56% vaccine efficacy for those who acquired it. So actually quite significant implications for interruption of viral transmission. You can see here in the figure, actually the primary outcome which was including all of the vaccinees, and comparing after the second dose, sort of the incidence of cases, the cumulative incidence of cases. And you can see also a quite good separation between the actual recipients of the vaccine versus the control arm, which in this case was actually a meningococcal vaccine, not just placebo. So you can see there was already a pretty good separation as well. And actually, after the first standard dose, similar to what the mRNA vaccines reported, you can see actually pretty good separation as well. So overall, interestingly, very good results, not as stellar as the mRNA vaccines, but certainly pretty good as well. Now, recently, actually, AstraZeneca released top-line results for the United States phase 3 results. Basically announcing that they met primary efficacy endpoints here in the United States as well. And they basically had over 30,000 participants, and they reported a 79% vaccine efficacy at preventing symptomatic COVID-19 and 100% efficacy against severe or critical disease, including hospitalization. Now, the comparable efficacy results across all ethnic and age groups, with 80% efficacy in participants over 65 years of age and older. So pretty significant and very positive results. Now, there's been some confusion about these results. In fact, actually, the FDA came and commented that this actually needs to be further elucidated, but certainly positive results overall, but perhaps needs to be further clarified. Moving on to the Johnson & Johnson vaccine, also known as the Janssen AD26 CoV-2S, or the AD26 is for adenovirus 26. This is also a replication incompetent adenovirus vector with DNA expressing a stabilized spike protein. Now, one of the major positive aspects of this vaccine is, unlike the mRNA vaccines in the Johnson & Johnson, it's actually only one dose. So you're done with one injection and you don't have to wait and for a second dose. So all sorts of logistically positive aspects for this vaccine. So they had almost 44,000 participants, all of which were over 18 years of age, a median follow-up of 2 months as well. Again, standard study design from the Operation Warp Speed. Included sites were mostly in the United States, South Africa, and Central and South America. And participants had several comorbidities, including diabetes, hypertension, heart disease, as well as kidney disease, including HIV-infected individuals as well. Overall, safety was quite high as well, including among those who were immunocompromised, like with HIV infection. Now, we don't have a publication on these. These were actually materials submitted to the FDA for approval. So you can see from figure 23, the most frequently reported systemic adverse effects in the study. And you can see that actually there were not that different or significantly different or serious compared to placebo. Most of it was fatigue, headache, myalgias, and nausea, most of which have been reported already. And in terms of efficacy from this particular vaccine, they were looking at moderate to severe illness 14 days after the injection. So the placebo arm had 348 cases, whereas the actual, the vaccine arm had 116 cases for overall efficacy of almost 67%. Now, for severe disease, that efficacy was higher at 85% efficacy, starting 7 days after its only injection. And actually that efficacy went up over time, up to 92%, 42 days after the injection. There were no COVID-19 hospitalizations in the vaccine arm as compared to 21 hospitalizations in the placebo arm. So that was 85% efficacy overall. And no deaths that were related to COVID-19 in the vaccine arm, as opposed to 5 deaths in the placebo arm. And you can see here from figure 12, the cumulative incidence of PCR confirmed moderate to severe critical COVID-19, at least 1 day after vaccination up to day 126. And you can see, much like the other vaccines, sort of a pretty significant and early separation between the vaccine arm and the placebo arm. And in this case was shortly after 14 days of the first or only injection for the Johnson & Johnson vaccine. So again, pretty significant and positive results. It led to the authorization of its use here in the United States shortly after the submission. Now, the last vaccine that we'll be talking about is the Novovax vaccine. So this is actually technology a little bit more familiar to us. This is a protein-based vaccine, much like the pneumococcal, meningococcal vaccines that we have been using for decades. What the protein that they use, much like the other targets, is the spike protein. So this is a full-length pre-fusion spike protein plus an adjuvant. So the protein vaccines are not as immunogenic as the actual RNA vaccines. So in order to compensate for that, you need an adjuvant to sort of increase the immunogenicity. So the Novovax vaccine is also 2 doses, 21 days apart. Now, one of the things that are important about this vaccine is also quite stable at refrigerator temperatures, no deep freezing required. They announced their top-line results on January 28, 2021. We're yet to see a full paper on this, but essentially it was a phase 3 study in the United Kingdom with over 15,000 participants. Again, the outcomes were PCR confirmed symptomatic COVID-19 individuals at least 7 days after the second dose. And the placebo arm had 62 cases, 1 of which was severe, and the Novavax arm had 6 cases, 0 of which were severe. The vaccine efficacy overall was 89%, with a pretty good confidence interval. We don't have further information for that vaccine because we don't actually have a FDA submission and further documents are lacking, but probably more that will come soon. Now, how do these vaccines translate in real world experience? We actually don't have a lot of data on that. These are all, as you can imagine, these were studies under optimal circumstances. However, we do have one that was actually quite a significant result that was published in January of 2021. So this was a paper that came out of Israel, which was an early leader in the vaccination efforts with quite high vaccination rates among its population. And this is data from its largest health organization, which used primarily the Pfizer vaccine. So they compared the Pfizer vaccinees versus matched non-vaccinated individuals. And actually, remarkably, were able to actually follow and have data on over 500,000 patients in each arm, so over 1 million people that they were able to look at outcomes. So included several categories in terms of comorbidities, including actually heavily immunosuppressed individuals in solid organ transplants and other individuals. So they were looking at efficacy 7 days after the second dose, sort of keeping similar to the phase 3 trials. And in terms of documented infection, meaning PCR confirmed infection, the efficacy in a real world scenario was 92%, so very similar to the phase 3 Pfizer vaccine results. Symptomatic COVID-19 was 94%, hospitalization efficacy was 87% protection, severe disease was 92%, and death from COVID-19, and this was a little bit different because it was before the second dose, they didn't have the complete data from that, it was 72%. So all in all, quite remarkable results in a real world scenario that the vaccine was doing what it was found to have done in a phase 3 study. And you can see here in the figure, sort of documented SARS-CoV-2 infection, again, a quite remarkable separation by day 14. Symptomatic COVID-19, similar. COVID-19 hospitalizations, actually even better. At some point between 7 and 14 days, you could see a difference. And then severe COVID-19 also by day 14. The death due to COVID-19 also, although the data is not robust here, pretty significant results as well. So this really led to a lot of confidence in these vaccines, a string of positive news about the results of these vaccines. And in fact, actually, shortly after these studies, the CDC was able to give some recommendations for those fully vaccinated individuals. So remember that the definition is 2 weeks after receiving the full dose series, if it was a 2-dose series for the mRNA vaccines, or 2 weeks after receiving a single dose vaccine in the Janssen vaccine. So what these recommendations basically have said is that fully vaccinated individuals can now visit other fully vaccinated individuals indoors without having to wear masks or the requirement for physical distancing. So quite positive news there. They can visit unvaccinated people from a single household who are at low risk for severe COVID-19 disease. They can go indoors and without wearing masks or physical distancing. I think a lot of grandparents were really celebrating this particular recommendation. And in fact, even for healthcare workers and other individuals, you actually could refrain from quarantining and having to undergo testing if you had a known exposure and if you remain asymptomatic. So for healthcare workers, you had an exposure, but you're fully vaccinated, you don't need to be quarantining or further testing. Now, what these recommendations also highlighted that everything else stayed the same. continue to take precautions in the public, like wearing a well-fitted mask and physical distancing, wearing masks when visiting unvaccinated individuals who are at increased risk for severe COVID-19. So those, for example, with advanced age or comorbidity. So a little bit of a, sort of nuanced recommendations, in particular when it comes to grandparents. But otherwise, you can see here that everything else kind of remain quite the same. These are subject to change. I think these will be updated over time as we have more and more data on the effect of these vaccines in the real world scenario. Now, additional recommendations and questions that have come up regarding these vaccines, they are not interchangeable. If you start with the Pfizer, you need to finish with the Pfizer. You can't be interchanging them. There is a minimum interval of 14 days before and after administration of any other vaccines. For example, the shingles vaccine, the flu vaccine, or pneumonia vaccines. Those obviously are still important to get, but you need some separation with the COVID vaccines. They're only approved for adults only. The Pfizer is approved for those over 16 years of age. The others are approved for those 18 years of age and above. There are a number of studies now underway in children. So this is an important aspect. And in pregnancy as well. It's likely thought to be safe, but really there are very limited data in this aspect. So studies are ongoing there as well. The duration of protection of these vaccines are unknown at this time. So if someone develops COVID-19 after full vaccination, it is important to follow standard recommendations in terms of treatment, similar to those who are not vaccinated. If someone's currently sick with COVID-19, what do you do about getting vaccinated? Those with symptoms should wait to be vaccinated until they have recovered from their illness and have met criteria for discontinued isolation. We certainly don't want someone who is thought to be contagious waiting in line. And if someone was previously diagnosed with COVID-19 and have recovered, by all means, they should get the vaccine, and it's still a strong recommendation. However, there is a thought to delay until they are no longer contagious, as you can imagine. And then if those who received monoclonal antibodies or convalescent plasma, because it's thought that those are quite protective as well, to delay 90 days until you require vaccination. Now, this is a recommendation in the setting of not having enough vaccines. Once we have plenty of vaccines, this may change. And then if someone is infected with COVID-19 in between doses, a situation that certainly has come up, the recommendation is to obtain the second dose after recovery and no longer thought to be contagious. Now, switching a little bit to a final component of this talk, we certainly have heard the news over and over again about these variants, sort of the UK variant, the South African variant, and then the Brazilian variant, causing quite a lot of concern to healthcare specialists and the CDC. So what are these variants? So as you know, viruses are constantly mutating, and this is nothing new about SARS-CoV-2, but there are some variants of particular concern. The UK variant in particular is thought to be more contagious and potentially more severe, and it has a worldwide spread at this time. The South African variant B.1.351 is also spread worldwide, and its major concern is there's a particular mutation in the spike protein that actually is able to evade some of the antibodies. So looking at the spike protein from the top, this is the receptor binding domain, and this is sort of some of the mutations that have been discovered in some of these variants, some of which render it more resistant to antibodies that were formed to the original SARS-CoV-2 or even to these monoclonal antibodies. So certainly something of concern. And then the Brazilian variant P1128P1 was first found in travelers coming back from Brazil and now subsequently discovered there. So far, limited spread. It's a close relative to the South African strain with some very similar mutations. The issue there has been the concern for reinfection. So there was this report at the Lancet a few months ago, the resurgence of COVID-19 in the city of Manaus in Brazil, despite high seroprevalence. So Manaus was a city that was heavily hit by COVID-19 in the spring, or there in Brazil was the fall of 2020, April and May, with really high rates of infection. And the population was actually, in a subsequent study, almost 76% of the population in that city was found to actually have antibodies to SARS-CoV-2. So that was a thought that maybe there was herd immunity in that population. And in that city, they were protected against further infections. But in fact, not what they saw in December and January of 2021, in which there was a second lockdown because cases were surging once again. And what they discovered was that the virus that was infecting those individuals in that city was the P1 lineage. So quite alarming that despite high rates of seroprevalence, there was no herd immunity to a mutated variant virus. So this is quite concerning to the healthcare community as well. So what information do we have about these variants and the vaccines? So very briefly, the Oxford AstraZeneca vaccine, in fact, actually, they did carry a study in South Africa and had over 2,000 adults that were enrolled. And in fact, actually, the primary endpoint analysis showed actually pretty poor outcomes. So there were 23 cases in the placebo and 19 cases in the vaccine arms who developed mild to moderate COVID-19 for an efficacy of 21%. And when you look at efficacy among those who actually acquired the variant, the South African variant, the vaccine efficacy was actually 10%. So this has led essentially to the vaccine being pulled from South Africa because of its poor results. And in fact, actually, when they looked in vitro neutralization assays, you could see a marked difference between the actual original vaccine and the mutated South African vaccine, and really poor outcomes there. The Johnson & Johnson vaccine actually also had a trial in South Africa where 90% of the strains were found to be the variant strain. Overall, the effectiveness was 64%, so a little bit more positive there, especially when it's compared to 67% overall and 72% efficacy in the United States. So not that different. So this was sort of hailed as positive outcomes. And 82% efficacy against severe disease, at least 28 days after the vaccination, and not that different, again, from the United States efficacy results. So, and actually there were no deaths in the South African cohort. So overall, the Johnson & Johnson vaccine perhaps still maintaining some protection against some of these variants, unlike the Oxford AstraZeneca vaccine. Novovax also released some study results in which in the United Kingdom, actually, the efficacy was quite similar, whether it was the variant or not the variant. But in South Africa, actually, the vaccine efficacy did drop to 60%. And in fact, actually, when you included HIV individuals, that efficacy actually dropped to 49%. So not as good there. And again, highlighting that perhaps the UK variant is certainly thought to still be susceptible to the vaccines, but perhaps not the South African strain. And then what do we know about the mRNA vaccine? So we don't actually have any clinical data, but certainly we have some neutralization assays. So this group engineered actually SARS-CoV-2 viruses containing the key mutations both in the United Kingdom, South Africa, and as well as the Brazilian variants. And basically what they found was that although the UK variant, which is here, and the Brazilian variant were actually not that different in terms of its neutralization activity in the vaccine, the South African variant certainly showed some decreased neutralization. So we don't know what the actual clinical implications of these results are, but certainly raising some concern that those vaccines may not be as effective against the South African strain. Now, in fact, actually, Pfizer has announced that they are working on developing a South African strain specific booster. So more to that to come in the subsequent months. The same results were actually for the Mrna vaccine, in which, again, we don't have clinical data, but certainly we have some in vitro data that they created some pseudoviruses expressing the variants for both the UK, the South African, as well as the Brazilian variants. And again, looking at neutralizing capacity, both from sera from human subjects as well as some non-human primates that received the Mrna vaccine. There were no impact in the UK variant, so good news there, but the South African and the Brazilian variants did show some reduced neutralization there. And you can see here, this is the UK variant, sort of not a lot of change in neutralization, but here, this is the South African variant, some reduced neutralization activity. So perhaps some concerns as well there that the mRNA vaccines may not be as efficient against the South African variants. And the reason, and Moderna, like the Pfizer vaccine, have announced a phase 1 study developing a South African variant specific booster vaccine. So again, more on that to come. Now, all of that in the United States, what's concerning to many is that actually these reports, actually even from our center here at Columbia, that we discovered a novel variant, B1526, that was identified in New York. They were looking at patients coming in with COVID-19 at various hospitals and actually identified very similar mutations to the South African and the Brazilian variants. And in fact, actually, those variants, or that particular variant that is now dubbed P1526, was rising at an alarming rate in mid-February. And in fact, it's, by March, it's up to 20%. So what's concerning is that this has some of the similar mutations to the South African variant, and perhaps might be less susceptible both to the monoclonal antibodies, but also to the vaccines. And now it seems to be spread throughout the Northeast as well. So we don't have a lot of data on this. I think we'll be watching this very carefully in the next months. So, and perhaps this is reflective. This is the latest cases as of March 2021. On the left, you have the United States. Certainly, we've recovered from that surge of cases in the winter, but certainly since beginning mid-February to March, there's been a plateauing of cases. So not coming down as fast as we would like. And whether this is due to the variants or not, we're not sure. But as many of you are aware, we're in a race against time trying to vaccinate as many people and perhaps preventing from new variants from emerging. And you can also see this worldwide. Similarly, from January, December, we've recovered from that peak. And actually, but some countries have seen a resurgence of the virus in Brazil and Europe, and that this is probably reflected here as well. So in summary, several vaccines are available in the US and worldwide. We have 2 mRNA vaccines and 1 viral vector vaccine that are approved already and being heavily used already in millions of individuals. The Oxford and Novovax vaccine candidates are not yet approved, but overall do show some promising results, in particular with the Novovax vaccine. There are several more additional vaccines in other countries, which we do not have to talk about, the time to talk about today. There's some early data from Israel showing positive real-world impact of vaccination in a country that actually did have a lot of the UK variants as well. CDC guidance on fully vaccinated individuals likely to undergo many updates, but certainly releasing some of the masking recommendations among those who are fully vaccinated. Unfortunately, infection rates remain quite high, and we're very concerned about the variants. There is really an urgent need for more data, in particular the vaccination response and these variants. But the expectation is that even if there are more cases, that these vaccines will still be protective, at least against severe disease. So we still have some hopeful data and expectations from those vaccines, even in the setting of these variants. And with that, I'll conclude my talk. Thank you, and looking forward to your questions.